When we first moved to Flock, we began probing around in our soils to get some baseline data on soil health, including its microbiota. Because as more and more of us realize, the key to healthy, resilient plants and ecosystems often rests on the health of our soils and the micro and macro community that live within the rhizosphere. Eventually, we tracked down Kyle Wickings, assistant professor of soil arthropod ecology at Cornell, who spends a lot of time analyzing soil invertebrates and their role in soil health to help us shed light on the secret lives of soil creatures.
I'm a soil animal ecologist here at Cornell, and my time is split between research and extension. So basically 60% of my time is spent conducting research, and everybody else out in the hall is basically doing all the research. The remaining 40% is extension, and I do a lot of work with the turfgrass industry and farmers trying to help them improve the biology of their soil health. So that's kind of what all the resources are around.
How did you get your start in this?
I started as a grad student at the University of Georgia at a long-term tillage trial looking at soil food web responses to tillage. So a lot of people get into soil food webs and soil health through till-no-till studies, and that's what I did.
Yeah, and how does agriculture in general affect our soil microbiology, and what are you finding from the top ground?
Tillage is probably the most important thing that we see that can reduce the size of the soil food web and its activity. So that's something that's been studied since back in the 60s and 70s. That's where a lot of it started, soil ecology really. So that's a huge one that can reduce basically the habitable pore space for soils. And we can sort of talk about what that is and how you increase it.
Yeah, I mean, because when I was doing entomology at Cornell, I studied more aquatic entomology.
Okay.
And then you look at like your biotic index of your water.
Yeah.
And if you have like certain...
We've got some similar things.
Yeah,
soil,
exactly.
yeah.
So I'm thinking like soil must be really similar because you're looking at maybe different species and the quantity of those species. Mm-hmm. And they could be relatively good indicator if your water is like polluted or stanky. If you only have mosquitoes, you're like, you don't have a diverse water system, and it's probably very stagnant and not so healthy.
So you probably looked at macro-invert shredders and
macro-invertebrates.
Yeah, so that's similar to what we try to do with soil food webs. We're always very jealous of the aquatic folks because they know way more about like indicators of stream quality, water quality in general, turbidity.
Do you have that kind of analog in the soil world?
We do. It's a lot fuzzier though. It's almost like tracing individual raindrops that contribute to a stream flow. You know what I mean?
I see.
It's like really tracking what each one of them does, how species differ in terms of their function is a lot harder to do.
And are you looking at both the microbiota and the macrobiota? So like,
Yeah, so a lot of what we focus on is trying to understand how small invertebrates interact with microbes. Because we sort of, even though we're fauna people, we think microbes really drive things in soil. But fauna, animals in soil, can really moderate that. They can change the size of the microbial community, their activity rates, and just the processes that they actually do. So it's sort of that interface between fauna and microbes that we look at.
Well, can you show us a little bit of like, I guess, what you look at on a daily basis?
Yes,
maybe
I'm going to have Hayden help out
are?
because we've got some really interesting samples we thought we'd show you guys. So
okay.
I don't know if you've got some stuff under the scope ready to show now, Hayden, but we can do that. We do have some live critters maybe we can show first. You could probably get them on that lens there.
Okay.
So one of the really common things we work with is columbella or springtails. I'm sure you've seen them before.
Yeah, especially when the snow starts to subside and you get the first warm day. We have a lot of those.
Snow fleas.
Snow fleas,
yep.
yeah.
They're a really unique group of columbella. So you'll see them eating algae and sort of the small plant matter inside the snowpack, which is pretty sweet. And as that melts and condenses, they're just grazing on it.
And then where do they go after the snow melt?
They live in the soil.
They just live in the soil, okay.
Yeah, but they're one of the groups, you see they're really dark in color, they're able to absorb sunlight and get warm in the soil profile. So, but this is a much different group from that.
Okay, they're not going to jump out at us.
They'll jump, but not be in our teeth and stuff. I'm sure I've swallowed a few of these. So what we're looking at is plaster of Paris around the outside. There's a little hole we cut in the middle, and we fill it with soil, and we feed them baker's yeast. Because they're microbivores, they'll feed on bacteria and fungi. And like the stream invertebrates you're talking about, they'll shred leaf litter too, but it's really to get all the microbes that live on it.
I see,
We call it the peanut butter on the cracker hypothesis. It's like you only really eat the cracker because it's covered in good stuff.
Right. And when we think about soil microorganisms, I think more in like regenerative agriculture or anything along those lines, they say, really feed your soil, but it's really feeding the things in the soil.
Yeah, so if you think about adding organic matter to the soil, you're doing that to build basically the physical stability of the soil, its ability to hold water, but it really is the basal resource for microbes and all these critters here.
Yeah.
So it's that fuel from the bottom up.
And it's not just like the archaea, the bacteria, the fungi, it's these guys too.
Yeah, so the thing about, if you contrast them, microbes are a lot better at breaking down organic matter. And we're actually looking at some assays upstairs right now to do that. We can show you guys too. But so they can produce all the expensive enzymes that break down crop residue like corn stalks and stuff. A lot of these fauna don't have that same kind of capability. They can do it, but they're a lot more dependent on microbes doing it. Or they'll let microbes they'll exploit their ability to break that down. And they'll either feed directly on the microbe or let the microbes sort of prime the organic matter and then they'll consume it.
So if they're just kind of at the tail end of this whole process, can you just have the microbes? Is that healthy enough soil?
Yeah, that's a great question. And if you look, if you sort of browse the soil health manuals, it really is very microbe-centric, focused on that. How do we quantify their impacts, quantify their basal resource pools? But what we're learning is that the fauna can really change the outcomes of microbial interactions and how microbial processes flow. So in that sense, they're kind of governing what microbes do and how fast they do it. So that's sort of the role that we see them playing in there.
Got it.
There's still a whole bunch we don't quite understand about it, but that's really what we're thinking. In a stream where you're thinking of them fragmenting the organic matter into smaller and smaller pieces, and it becomes part of the water column after that, we have sort of the same process going on with fauna here. They'll shred organic matter, they help take it from the soil surface down into the profile and help move it around, but they're also sort of driving that microbial engine a little bit too.
And I would imagine too, like some of the larger fauna in the soil, I would imagine this could serve as bird food to a certain degree.
Oh yeah,
know,
yeah, so
especially the ones that are pupating.
It's interesting because you have some soil scientists or soil ecologists that really drill into this stuff and really don't even think about these, and then others that just focus on this and couldn't identify a scarab at all. So,
so
trying to walk those worlds. These really are part of the soil food web as well.
That's right. Yeah.
They might be feeding exclusively on roots. Some of these species will feed on dead organic matter at one stage when they're larvae, and they'll shift over to feeding on live roots later on. So in various ways, they're becoming part and then leaving the soil food web. But all of these either have their entire life cycle or their larval stage or their eggs or pupae in soil at some point.
I mean, it's the same with most of the insects in water as well.
Like
the dragonfly, mayflies, storkflies,
most of their life below the water surface as a shredder and a stream invertebrate, and then hatch for a couple days.
And serve as fish food.
Yep.
Yeah, as well.
Well, we could do a whole section on fly fishing. So this species is called Isotomiella minor. It's a really common columbella here in New York State. We find it in lawns. We find it very commonly in forest floors and stuff like that.
One of the other things that, when we were kind of conversing over email is that you said that, obviously during the seasons, maybe it's not so obvious to some folks, the micro and the macrofauna change dramatically.
Yeah.
Can you just share a little bit more about that? Because if you're taking at one point of the year and you say, oh my God, I have nothing in my soil, or I have very little, what do you see on a seasonal basis, especially in this area? Yeah,
it kind of follows plant productivity in some ways, but in New York in particular, in the Northeast, in spring and fall, we have a peak in fauna activity. If you look in the middle of summer, you're very likely to think your soil quality is just horrendous because you don't find as many fauna. And they respond in so many different ways to drying out in soil, to increasing temperatures. We actually exploit that for how we extract fauna, which we can talk about in a bit too. So.
Yeah, and another question I have is when I'm taking a soil sample, I'm going maybe 6 inches in, getting a little soil sample to test for whatever, heavy metals, pH, that type of thing. How are you taking a soil sample? I see ones that are here, but...
Total nerd shot, right? So most of the biota we see lives in and around the root zone. We call it the rhizosphere, the soil that's immediately in contact with the root. And some of that is super rich and basically covers the whole profile, like in grasses where you have roots, every millimeter basically, and others are very isolated, like in crop rows. So they're isolated kind of around that because all the organic matter that roots are pumping out, the microbes and the fauna are sort of using that as fuel.
Okay.
Basically rhizodeposits.
So if you don't have a deep root system. They're just really there on like kind of the upper.
Yep, they'll use leaf litter at the soil surface. They'll use that as a resource, but most of the carbon that ends up in soil is coming from roots being input below ground. So the spatial dynamics of roots really drive where the biota are going to be.
Now, when we think of like earthworms, which I'd like to actually touch upon later, and you're like digging in the soil and you're like, ooh, you know, I have a worm going down. Are there ones that actually are like the pioneers, like going out west, young man, you know, do they go out like down into the soil surface even further than the roots?
Yeah, so a lot of earthworms will take organic matter from the surface and take it down into vertical burrows. So they'll be taking stuff from here and sort of translocating it below ground. So in that sense, yeah, sort of the depth pioneer, right?
Yeah.
Especially in a compacted soil that hasn't had biota for a while in it.
Yeah.
And those pores are also now highways for other biota, for water, for plant roots, so they're really engineering that soil profile.
Great.
But most of the fauna, like these critters, they're right near the soil surface in the top a few inches of soil. Like, so when you're saying you're taking a 6-inch sample, you're getting most of the zone where the biota are most active.
And is that also part of the reason why, you know, when you flip soil,
Like
Kirsten,
plowing.
yeah, Kirsten Kurtz had mentioned it to me, who's a director of or was working with the soil health lab at Cornell, and she's like, you know, it's kind of like if you have the penthouse suite and you have the first floor and you're kind of switching it around, and they live in certain areas.
Basement's on top now.
Yeah, exactly.
Yeah, so that physical disturbance is huge for fauna. How they differ is in how quickly they can recover from it. Some, they really thrive. When you cultivate a soil, you burst open aggregates and free up very labile carbon, and a lot of things will kind of bloom beyond that. So they can deal with the disturbance, their eggs survive it, and then they'll hatch. And you'll see spikes in their density. You can almost tell that was a tilled soil when these fauna show up. Others, like Collembola, they're affected pretty heavily by tillage, but they can recolonize that soil pretty quickly.
Okay.
And then others, like mites, which we'll show you a bunch of those in these samples, they're much slower. And even after 10 years of heavy cultivation, you really don't see their communities restored the same way.
And when we think about mites, sometimes people think of like, ooh, mites, you know, ugh.
Yeah.
But some of them are probably good and bad. You get both beneficial and non-beneficial, I'm assuming.
Absolutely. And in soils, you know, we do have some pest mites, like spider mites, for example, that are more at the soil surface and on foliage. That can be really bad crop pests. But most of the ones that we see are either decomposers or predators. So.
What about
there's like 50,000 species of them. So.
What about nematodes? Because I know that there's like nematodes are maybe the most prolific organism on the planet.
Have you ever heard the story of you basically replace your entire body mass with the nematodes right below your feet?
No, I had no idea.
It's huge. They're
crazy.
Incredibly abundant in soils.
And nematodes as well, like both beneficial and non-beneficial. You get
same scenario.
Yeah.
And some of them, in terms of the sort of pathology side, are horrible crop pests as well.
Yeah.
So that's actually a huge educational piece when we talk with producers and turf managers, because they think, man, I've got these horrible stories about nematodes as pests. So you're talking to me about them, you want them in your soil and all this. So distinguishing beneficials from pests is tricky, but also really important. So we work with nematodes as insect pathogens. A lot of them can colonize, kill, and eat insects. So we use them in biological control. And there's a whole range of free-living nematode species as well that are predators, fungivores, decomposers. So yeah, they're really good indicators of soil health as well.
Well, should we take a look at some of these mites?
Yeah.
And are the mites predatory or beneficial?
There's a mix, and we can actually get cameras through the eyepiece pretty well. I don't know if you want to try that, but to do it. But
ooh, look at everything under there.
Yeah, let me get you oriented there. And so this is actually, it's kind of skipping ahead to the earthworm part, but we're doing a project to see if the invasion of an invasive species, the jumping worms, impact soil biology in the Adirondacks. So there's a lot of areas where jumping worms...
Are they in the Adirondacks now?
There are areas where they are, and there's a number of areas where they're not.
Okay.
So we're comparing areas with European earthworms, jumping worms, and no earthworms to see what the impacts are.
When did... we're jumping ahead.
Yeah.
So I don't want to get too much into the worm.
So I don't actually know whether this is a jumping worm or non-jumping worm sample, but it's a real rich one. So this sample basically came out of this material here, and there are just thousands of critters that you're going to see.
Well, take a look at that. See if you could get the camera lens in there. Yeah, look at that guy.
Zoom out a little bit so you can see the whole thing. So what we're looking at here is a mix of what we call macro-invertebrates and mesofauna, or microarthropods. So you're looking at this. So that big critter right there, that's a millipede. So we call that a macroinvertebrate. They're like the shredders in the stream. So they're the ones that are fragmenting this material up, kind of breaking it down so it moves into the soil profile. And then all these other critters that you see around it, we call those microarthropods.
Did you just like shake some leaves out? Like how did you just...
So we'll show you the extractors of how we get these. This is
Okay.
actually a really good school activity as well.
Yeah.
Because anywhere you can find numbers like this, it's pretty wild.
Yeah,
So
it's crazy.
let me see if I can just
So
point
like you
at a
said, you don't
couple.
know whether this was with the jumping worms or not.
We don't know yet. We're trying to keep that blind to us at this point, so we doesn't bias any
That's
of our
smart. That's
sampling.
smart. And I see some colembula in there.
Yes.
Like right there.
Yeah,
and-
I'm going to try to point at one without totally screwing this sample up. But so right down here.
Yep.
Oh, yeah, that's going to mess it up.
Yeah.
Right at the top of that,
Yep,
little
that
long
little
thread-like
white-
thing.
yeah.
That's a springtail. You can see some of the dark brown things, the little globe-like things. Those are called orobatid mites.
I mean, they almost look like little ticks.
Yeah, so they're all related. They're all
Yeah.
in the acari or in the arachnids, and this is the acari or the mites. So some of them look very tick-like. Most of the ones you see in this image here are orobatid mites. They don't really respond to disturbance super well, like talking about cultivating or plowing soil.
I see.
That's one of the quickest groups you see drop off after that. So another good indicator that you've sort of restored the physical aspect of your soil when
So
you have a good richness of
Are
these
you
guys.
basically counting like how many species that you see in one section and what types of species, and you're just classifying them? And can you classify them down to species, or is it just genera at this point?
Yeah, so the keys that we have, they're pretty tricky. Some of them you can't, unless you have a female specimen, you can't actually identify it.
Okay. Yeah.
But what we do is we'll break them down by something like suborder. So we can say orobatid mite, mesostigmatid mite, prostigmatid mite, very, very coarse taxonomy, and then we break it down from there. The challenge with that is you have to go from this scope to a slide mount to get it beyond something like the family level.
Yeah,
And oftentimes to get
okay.
it to family, you need to put it on a slide as well.
I just
So that's a
remember
real challenge
Having
deciding.
to count hairs and pores and things like
Yes.
that.
And this is all about, we call it ketotaxy, or where the hairs are, how many, on
Yeah.
what section of the leg.
Right.
So it can take a sample like this with thousands of microarthropods in it and make it, dozens to maybe 100 hours at a scope to get everything identified.
Unbelievable.
So I don't know how much zipping around you want to do, but those white binders in the back, those are the keys just for figuring out soil mites. So,
My goodness.
and this is why there aren't a ton of people that still do this.
Yeah. I mean, how could you?
Yeah. So it's based on body hairs, and all are with the assumption that you've put it on a slide mount. So great books, really, really good courses, actually a summer course on mite identification. But yeah, so if you're asking an ecological question and you need to know who's there, you have to really quickly decide how far am I going to have to identify everything to figure
Yeah.
out its function.
Yeah.
So we tend to walk the line between taxonomy and function. We'll identify coarsely, call them morphogroup whatever the whole time, and then at the end we'll go in and do some slide mounts from each vial and figure out who's who, to at least get them to family, so we can talk about both taxonomy and the ecological role they play.
So basically what you're saying, just so I could summarize, is that certain sorts of micro and macro fauna in the soil, especially when you're like tilling soil or disturbing it, it changes dramatically, and that could change dramatically the amount of organic matter or the soil health or aeration, all that type of stuff that's related back to soil health and plant health.
Yeah.
And
yeah.
then some take longer to recover and some don't. And you might see that in plant health, because most of us are not digging in the soil and like spending hundreds of hours looking at like counting hairs on like mite legs,
Yeah.
for instance.
Yeah.
And then I guess my question is like, how much disturbance is needed in order to be able to really affect a community? Because I know in forestry sometimes they tell you to like go in there and scuff the soil surface and everything so you
Yeah.
could actually get forest regeneration
Yep.
and things along those lines. And of course, like when you're putting in even a bare root plant, you're digging in the soil to some certain degree.
Yeah.
And
That's a great way to look at it, because you have to kind of soil disturbance is part of the process of a lot of biotic interactions in soil.
yeah, I'm thinking like even bears scuffing up
Yep.
the soil or something digging up.
Earthworms engineer the soil,
Yeah.
so it's definitely happening, and it's an important part of like the stability of soil building.
Right, and I noticed like a poster that was out there about like your effects on walking on soil,
Yeah.
and you're also doing something on mowing. Like how
Yep.
does it actually affect turf grass by just simply mowing? Which I know we're in May right now, and there's like the no mow May, and we
Yeah.
don't actually mow until June, and then we
Oh, great,
don't
yeah.
mow until September,
Yep.
June and September, and we try to just only mow paths, and the rest is kind of like undisturbed. how little disturbance do you need to do in order to not affect, like the optimum soil health, I guess is what I'm saying.
That's a good question. You know, I think some effects we'll see even just from one, say, especially if it's like a moldboard plow where you're really folding the profile over. And that tends to come from the immediate, just like you're actually crushing and killing critters. But beyond that, the effects happen from changing the pore space. So this super nerdy drawing we have over here. If you picture the soil profile from the side, these pore channels are really what's important for biota to survive and move, find resources. So when you're moldboard plowing, you might be killing things just with the immediate impact, but you're also crushing aggregates, you're breaking those aggregates down, and you're shifting these pore spaces to be smaller and fewer. So you're just taking that habitable pore space for fauna and reducing it considerably. It's like if you made all the hallways in your house like a quarter of the width, right? It's just gonna affect a lot of your movement.
Yeah, let me ask you another question, because I'm gonna approach this from a plant perspective. And in kind of the old school thinking, you used to plant a tree and you used to be able to amend the hole. Like say you have really crappy soil, like it's just full on clay or whatever, and
nothing for roots to penetrate.
Nothing for roots to penetrate. And now, you know, kind of the school of thought is actually, no, you only put that compost and the mulch on top of the soil and you leave, you know, what's in there. But if your soil is kind of like wrecked, would it be better to like amend it and put some leaf mold in there, and then, you know, could you reintroduce these species sooner rather than later, or is that just not copacetic?
Yeah, no, it's actually a really good practice. There's lots of nuance to it, right? Like using the right material, something that breaks down fast versus slow, and there's a lot of debate over like what you actually need to do to promote recovery of soil biota. So it's not really an exact recipe at this point, but we know that it's increasing their resource base, and all those things add to the way soils retain water, the nutrients they provide, and just the physical structure that soil maintains. So that is a way you're restoring the habitat and the resource base of soil.
So meaning amending in the hole or out on top of the hole, or both?
Oh, I would say if it's a tree, we're talking about that. I think probably that practice of amending within the hole, like around the pot set that goes in, is probably still a good common practice, just to establish the roots, allow them to actually move out.
Yeah, fascinating.
So how that blends though with like the recolonization of that root ball by fauna, I don't think we actually know that. That's a good question.
Okay.
But
These
I would say,
are all the questions I want to know.
I would say if you promote a healthy root system for that tree, you're going to get a lot of recolonization, because that rhizosphere is really what they're dependent upon. So.
What are you also testing as far as soil health and quality outside of this? Like, do you go back to the soil health lab and say, okay, what are the heavy metals? What are the other things in here to kind of understand the impacts of that on there? Because, you know, I actually I was asking Kirsten from the, you know, soil health lab if I could do more extensive tests than even that they offer, finding just certain things like within, just because I want to have a nice solid baseline of the soil that we're working with.
Yeah.
And kind of understanding what's in our environment, like the area that we're planting now used to be an old container nursery for 30 years. They probably put in a lot of pesticides and herbicides and all that other kind of stuff during that time. You know, they removed the topsoil. They put, you know, 4 to 8 inches of gravel on top of that. And I'm like,
history of the soil is like
the history
crazy.
of the soil is like really intense, and I'm interested in getting a baseline and seeing like, are our practices actually doing anything whatsoever? Are we even seeing like animal life in there at some point? So what are these other things that you're studying in order to kind of sync up with the health of the soil?
So we do look at physical traits of soil as well as microbial. We have a lot of toys around the lab just to measure microbial fractions and their activity. So I think that's a really important indicator.
But you haven't looked at like herbicides or pesticides or
we
certain
do,
things like that?
So we have a lab on main campus that we can quantify pesticide residues.
Got it.
And we'll look at that from a couple different angles, trying to help make managers that are reliant on the pesticides more efficient with their use. Like how much is still in the soil versus been taken up by the plant is a really important step. So that's one thing that we do as well.
On a related level, sorry, I'm peppering you with all these questions, but it's actually relating back to some of the other things that we're looking at and filming. Hemlock woolly adelgid. Right now in New York State, there's only funding in order to do pesticide application in and around the root base of hemlocks.
With probably the neonicotinoids,
Yeah, with the neonicotinoids. And I know that some of the questions that people had on some of these Zoom conversations and Zoom presentations were like, how does it affect the surrounding soil health now and into the future? Does it actually like kill a certain thing? One of the things that I had read is that it kills a certain amount of things, but because it's an insecticide, it doesn't actually affect the mites, and the mites explode.
Interesting.
And I was like, okay, well, if this is what's being promoted to citizens with their hemlocks, what are the things that we don't understand or that we haven't actually looked at from that standpoint?
Great question. So we've seen the same thing. We haven't really seen the blooming of mites following the imidacloprid or neonicotinoid applications, but we do see really short-term effects on columbolans. But so they will tank really quickly. We see them recover fairly fast from the products, though. But it can have. It's just because it's an insecticide doesn't mean it can't impact the microbial community as well. So far, it seems like the recovery is fairly rapid with the fauna from that. But, and as you probably know, there's a whole bunch of restrictions on who can use neonicotinoids now and where in New York State. So they're really not available even for the residential market right now. Like a homeowner couldn't get it, for example.
Yeah, exactly. You have to get somebody who's a certified pesticide applicator in order to be able to do it. So exactly. It's another hurdle. Fortunately and unfortunately, it seems like another hurdle for somebody to actually do something with their hemlock.
Yeah, there's a lot of interest in using biological control like that. We use it in soils. Nematodes, in fact, we'll use to kind of control white grubs. The challenge is you're buying or cultivating a live organism. You've got to keep it happy, introduce it right so it can actually proliferate in the soil, find its host.
But they also think the thing that I think you probably hit home on is don't do that thing in the soil in the first place that promotes, the white grubs or something along those lines. But I guess you can't control that, especially if you have a golf course, for instance, and you're mowing all the time, for instance. That's going to be a thing that is what it is, and you have to kind of work with what you have.
They really love grass. I mean, grass roots are like the perfect food for these things. They're moist soils, so they can colonize it well. We still don't know exactly what drives the patchiness of something like scarab beetle larvae in soils. We're still kind of working on that, like why here exactly. But we know that most of these sites don't have high enough background of natural enemies to suppress their populations. So one of our goals is to help just better decision making about insecticide use. So use them exactly where you know they're going to count and when they're going to count, and that alone can reduce pesticide inputs dramatically. But it takes a lot more decision making. Learn how to scout, learn how to identify grubs, and know the timing.
And what are you finding for homeowners that homeowners can do, like in their own backyards? What kind of practices can they do, or gardeners even?
I think with homeowners, I guess it relates to suppressing pests, but also improving soil health, aerifying soil, trying to traffic it as little as possible so you don't really compact the soil. There's a lot of shift that we're seeing right now in perspective of homeowners and what they want out of their grasses. Some still want mostly aesthetic and cultural value. They want it to look good, they want it for picnics, parties, all of that, and they want people to traffic it. But we also know that there's general interest in its ability to retain water, prevent erosion because of all the roots that grasses have. And more we're seeing people want to get more ecological value out of their lawns. They want to either host pollinators and other wildlife. They're thinking about sequestering carbon because we know that grasses actually are really good at sequestering carbon depending on how they're managed. So those conversations are popping up all over in home lawn management. So thinking about, well, how do we balance these cultural expectations with some of these other services that people want to get? So that's like a huge area of research for us right now. And it always involves soil biota, because if you're interested in biodiversity in lawns, that's where your biodiversity potential is highest.
Yeah. And you'd be taking some of the biodiversity, you'd be testing every season, or how do you test like one piece of lawn to make sure that it's healthy? Because like you said, in the summer, it looks like it tanks. Or are you only taking things in spring and fall?
We typically will do our sampling then. If you have times in summer where you have some adequate moisture, most of the turf we sample is not irrigated turfgrass. So Hayden's doing some work in Rochester City parks and just seeing huge levels of diversity there. But we'll typically hit the sampling in spring and fall for that.
Yeah. See, I'm just really curious about like our practices on our own land and just like mowing less. Like we don't have really a baseline of what it was, then and what it is afterwards.
Yeah, trying to envision what is your end goal with a community like that, a plant community. So there's people interested in completely renovating, ripping out, which is going to require some disturbance, and planting whatever assemblage of native plants they can in there. But there's also people that just want to just manage less intensively. Mow it, forget it, try to raise the mower deck up as high as possible and let it be that way.
It never is, you cannot raise the mower deck up tall enough.
Not on most mowers now.
No,
yeah.
we need something that's at least like 6 inches.
I think there are companies that are interested in looking at that because they know people want to raise the deck higher.
Yeah.
Well, there's a problem because I think the mower creates a vacuum.
Yep, with the deck
lower
height.
it is to the ground, so the higher you go, like if the blade comes around, it's just the grass is just gonna fall over and then dodge
won't
the
eject
blades.
the clippings the right way, and
so
that's really the engineering.
But that's an
challenge
engineering challenge.
Yeah.
I think that it could probably be solved, but maybe not the same way that you've built mowers before. You know, that was one of the things that we lamented at, and we're like, you know, on this wheelbarrow with like a hedge trimmer, and we're like pushing down the
faster.
We're trying to like
I'd like to see that.
cut the lawn, but we've done it all. We were like, we've ripped up the sod and put bulbs in and put new sod on top. We've done you know, pollinator gardens. We're now like doing low mow in certain areas and letting it grow as is. We've transferred over to, we've reseeded with like low mow native lawn mix.
Oh yeah, yep.
And only like native lawn mix that you'd find in here, and the rest is like Eurasiatic grass. So I'm even thinking that Eurasiatic grass might have a different effect on the microbiota community than maybe some of our native grasses.
Yeah.
Like, can you find something that's in the native grass that you wouldn't find in the Eurasiatic grass? We don't know, right?
Yeah. Yeah, and I think a lot of that comes down to the root traits. So how much diversity do you have in the root zone, and whatever those exudates are or products the roots are like depositing in the soil that could be communicating with microbes and driving their responses. So those are all really, that's exactly the type of conversations that we've been talking about, like that homeowners are interested in. They want to know what to do. How do you be prescriptive about that and deal with all the variability you get when you try something?
Yeah, I think the key thing though is letting folks know that there is actually something underground.
Yeah.
Just because it's buried under there, it doesn't mean that it's not alive. And I think that's one of the crazy things, like even people in aquatic entomology, people didn't even recognize there's bugs in the water. And actually, yes, the types of bugs and the diversity of bugs and how many bugs you have actually might mean that the water's cleaner.
Yeah.
You should want bugs in your water.
And a whole bunch of things dependent on them too, you know. So I just figured, since we're talking about that, this is actually a sample from an urban park in Rochester.
Ooh, this doesn't look as
So there's a little isopod in here, but I'm just going to zoom in on the mites again. So it's pretty interesting because even in a park, you can still get pretty high diversity of the critters that are in it. So these are mostly orobatid mites. So you can see some springtails in there, the long guys with the antennae. There's some predatory mites in here as well. So those would be feeding on columbolins, other mites. They feed on nematodes. So it's a mix of decomposers and predators.
Look how cool. It's just so cool.
In here.
Look at those big black ones with like funky mole-like legs.
Oh yeah. See, let me see if I can see the real leggy one, the tan one.
Yeah,
right
the
there.
tan one, yeah.
So that's that's a predatory mite.
And those probably have like big grasping legs, like, right? Because you'd have these.
Yeah, so they're actually
grasping
fluid
legs.
feeders. So what they'll do is they'll grab their prey and sort of jam their face into it and liquefy it and drink the contents of it.
Cool.
So, yeah. And then the one you were just talking about.
Yeah, the big mole-like one.
Right, yes, I'm sure everybody can see the biggest one in there. So that's another kind of orobatid mite called a golumnid. And they actually have shields that they can lower over top of their legs to protect their bodies.
Huh.
So.
What a different world, right?
Totally.
Wow.
They're very slow. They're kind of the tanks of soil animals.
Yeah,
so
they
you
look
can
like a
picture
little
how
tank.
they wouldn't recolonize as quickly as other things. And
Yeah.
so the predatory mites are a lot faster. Some of those predatory mites are the same general group of mites that are commercialized to sell for pest control in greenhouses, actually.
Huh.
So.
What are these little brown ones with the little black centers? You see those little black dots in there?
Yeah, the black dots, that's actually that's their stomach content. So they call it a food bolus, and they'll just sort of like condense around it. They can extract moisture from it, and they'll retain that bolus in the soil. We did a project where we looked at the chemical makeup of the food bolus.
Yeah. Wow, that's crazy. How do you even extract the food bolus? It's
You
like
need
tiny,
many of them.
Yeah, tiny and tiny.
So these
I already checked out this
good.
book.
Well, I
was
have a
like,
copy for you
soil
guys.
Animals,
yeah.
yay.
So these are Berlesi funnels. This is kind of a combination of a couple different things. There was a funnel called the Tellgren funnel and the Berlesi funnel. When Berlesi funnels were invented, they used hot water jackets around the funnel itself to try and warm the soil and dry it. Whereas the tall green funnel used light. And basically, in both cases, you're trying to dry the soil out and make a temperature gradient in the soil. So animals in soil, when things dry out and get warm, they go deeper in the profile. So we're kind of exploiting that.
I see.
So leaf litter, anything, old bird nests, you can put anything in these and extract critters out of them.
I mean, that's another thing that if you have exposed soil and you have no leaf litter on it,
you lose that buffering
You lose the
capacity.
buffer.
I mean, soil
Yeah.
itself has a really high buffering capacity, and that's why so many things use it for their pupae, their eggs, their larvae. But that surface buffering capacity really does drive a lot.
Yeah.
So that's why home gardeners, one tip they can always follow, just keep soil covered. Whether it's a cover crop
Right.
or plant residue is a really good way to maintain the buffering capacity.
Here's another question, again, coming from a gardener. I mean, a lot of the times Kirsten was mentioning about how you could flip the soil. What about when you put cardboard over the top of it, which we've done, and then we put kind of more soil on top of it, and so you're almost like burying them on the bottom.
Yeah.
Is that the same thing that you're doing? Because then when I look into that soil that I just have new soil and mulch, I mean, within days you start seeing earthworms, you start seeing, beetle larvae, you start seeing lots of different things in there.
Yeah.
So what did you do to the ones? Did you suffocate all the ones down below?
Yeah, good question. I guess if you really compacted the crap out of it, you could, but I
Yeah.
would say if you're not physically really churning it heavily,
Yeah.
they'll just recolonize and move into that part of the profile.
Okay, so they'd come up probably up from through the cardboard at some point.
Yep,
and then,
as it rots. Even during the day, fauna
Yeah.
you'll see this sort of diurnal vertical migration in soil. Just
Fascinating.
for resources, moving away from or following water
Yeah.
and temperature. So they exploit that gradient all the time.
See, I
and
wonder-
then we trick them to fall out into a...
I just wonder if they thank us from doing that, because there's a whole movement around like the no dig methods, and you just put cardboard on top, you kill the lawn,
Yeah.
and you put all this stuff. Like, I just want to know, are microbes thanking us below,
Yep.
saying, "Ooh, now we have more layers of soil to go in"?
It's a great question, and there's a lot of pride in gardening and like
Yeah.
mixing your soil, and it's like coffee grounds or something,
Yeah.
But it's similar to cultivating a soil. You're kind of plowing it. So trying to do that as gently as possible, but you do have to incorporate organic amendments. So minimizing it where you can, I think, just like with the conservation or no-till movement for commercial producers, is really important. So to keep those aggregate pore spaces present. So, but yeah.
Continue. Sorry.
No, that's pretty much it. So these lights come down on top. We set all these up. It looks like Christmas lights in here when they're all on. They're on a dimmer switch, so we slowly raise the temperature from ambient up till we can completely dry the soil out. And after about 3 days, we'll have the soil collected all into ethanol.
Wow.
We can collect them live as well if we want to use them for studies.
Yeah.
So, but this is sort of the sciencey version of this. There's a very good DIY version in this book that people can make at home. Producers, gardeners can set them up and just if they're going to amend soils aggressively one year, take some samples at the beginning, just put it on your kitchen table with your Berlese lamp, and keep checking it over the season to see, are you increasing populations? And you don't have to get down to the level of like, oh, I'm going to look at body hairs and count all these things. It's just general numbers and how diverse does it look is a good practice. So
This would be one of those subjects where it would really be hard to get citizen scientists to do this. 'Cause you also need like a microscope and everything along those lines. It's not like bird counts,
So let me show you one of the hand lenses we promote people use. Something like this little reading magnifier is just fine for it.
Is that like a 10 times or 20 times?
I don't even know what X this bad boy is.
It's more than a hand lens. It's like a...
It's for reading and looking at fine diamonds, apparently.
I don't think they wanted to put microbes on the front cover.
Yeah, like
that.
a bacterial cell.
Yeah,
I don't know why.
diamonds seem a little bit more like...
Freaking my grandma out.
Yeah.
But I mean, even with that, you can see a sample well enough to say, is this one more diverse than the last one? So let me grab the animal sample. Put that under there.
like increasing my pore size.
Do a nose hair count. But yeah, you can look at it through this and just get a good visual for it. So that's a pretty straightforward way to not feel like you need a microscope.
Right.
And just assess what's happening in your soils.
Well, I mean, thank you so much for sharing this world with us, because it's such an unknown world, and folks just don't have a clue. Like we hear about soil microbes, but we just are not put in touch with them. And just even looking through the microscope alone, it's just like a whole other world.
It's like that's down there.
Yeah, indeed.
Yeah,
well, thank you so
absolutely.
much. Really appreciate it.
Yeah, you bet.
Stay tuned here, because we'll have more stories from the underground world here on Flock. If you enjoy these educational videos, then do us a favor and give them a thumbs up, subscribe, and hit the notifications button so you know when new videos release. We work hard to bring you solid content to help you become better stewards of the environment and would greatly appreciate the love in return. 10% of our Google AdSense proceeds are reinvested back into community projects here in the Finger Lakes, and that's matched by our partners over at Espoma Organic, which is a fourth-generation, family-owned and operated business that has been on the forefront of organic gardening since 1929. Thank you again, and we'll see you in the next video.